Good afternoon and welcome back to Excelsior Academy. So today we'll be doing gene therapy, a last portion from your syllabus of animal tissue culture. So gene therapy, so it involves the introduction of one or more foreign gene into an organism to treat hereditary or acquired genetic defects. So this, as we were discussing, so already we know what is the meaning of introduction of one gene or more foreign gene. That is, it is the transgenesis, right? The procedure is called transgenesis and the animal will be called as transgenic. Now in gene therapy, DNA encoding a therapeutic protein is packaged within the vector. So what we are doing is, this is just like cloning only. So cloning means you have a vector. Okay, so this vector can be from the bacteria or it can be from the virus. To that vector, you are adding your gene of interest. And finally, this will be transferred into the host cell so that it can have or it can show its function. Now, what are the medical conditions in which gene therapy has been studied? So first one is ADA, adenosine deaminase. So it is responsible for SCID. So this, as we were discussing before, it is severe combined immunodeficiency, right? It is immunodeficiency. So in this problem, in this case, this enzyme called adenosine deaminase is deficient. It means it is not functioning. So what gene therapy is doing? So in case of gene therapy, the doctors and the scientists, what they have suggested is taking the bone marrow, okay, of a healthy patient, introducing the ADA vector, and then introducing into the patient. Okay. Now the problem with severe combined immunodeficiency is that they are very susceptible to anything. I mean, susceptible to all the organism. For us, it's like we are resistant to many airborne sorry, infections. So these people are very sensitive, very susceptible. So once they are born and suppose they are diagnosed with this kid, so they are always keep under the bubble. Okay, if you see, bubble means the inside the plastic chamber they will be kept, and sterilized air will be passed for them to breathe. Okay, then the other medical conditions is liver, brain tumor, muscular dystrophy, diabetes. Insulin gene has been, they have tried, transferring using the vectors. Prostate cancer, hemophilia, asthma, melanoma. So mainly it is, if you see, it is all cancer, right? So mainly the cancer is being targeted over here. Now, as far as I remember, even in my hospital also, we were using it's not exactly the gene therapy case, but we are always targeting the cancer only. Because the problem with cancer is that the moment you find one drug which is able to treat one particular cancer, after few generation of the cancer, what happens? It becomes resistant to that particular drug. That's why till now we are not able to cure cancer. Okay. Now, what are the types of gene therapy? So, somatic gene therapy and germinal gene therapy. So, from the name itself, it's very much clear. So, somatic gene therapy means somatic cells of the patients will be targeted for the foreign gene transfer, and it is not inherited. That is, it is restricted to that particular generation only. It will not be transferred to the other generation. Whereas in the case of germinal, it is present in the germ cells. So, of course, if it is targeted, then it will be giving rise to the, it is passed to the offsprings also. Okay. Now, gene therapy approach. So, classic gene therapy, it involves therapeutic gene delivery and the optimum expression once inside the cell. So, what is happening? So, you have got the gene. Suppose this is your vector. This is your interested gene. This will be transferred inside the host cell. So they're inside the host cells. Okay. Now the thing is that it is important to understand that there are two ways. If it is inside, whenever this is getting replicated.
Ma'am, record.
Yeah, it is showing recording.
Now we are.
Now,
Yes, ma'am.
okay. So there are two ways. So once this is getting replicated, of course, it is going to show its expression. Okay. Now, gene therapy doesn't mean that it is going to be always producing the protein expression. It can be RNA also. So whenever we say gene expression, it can be either in RNA expression wise or in the protein case. Okay. The other ways is we are just targeting over here and it gets integrated inside the genome of the host. And then it is getting replicated, giving the expression. So these are the ways by which a gene gets targeted. Okay, now non-classic gene therapy, it involves the inhibition of expression of genes, which is associated with the pathogenesis or to correct a genetic defect to restore the normal gene function. So classic gene therapy means there is a defective gene. In order to, for that gene to work, you're inserting another new gene, right? And in certain cases, you just don't want the expression. So you're going to do the inhibition. Now, how you're going to do the inhibition? Remember from the previous class, how do you inhibit the expression of a gene? What is it?
Knockout.
Very good. So you do the knockout of the genes. Okay, so that also will be done with the help of the vectors. Okay, so the foreign genes may carry out the following function. So it can produce the product in the form of protein, or it can be RNA also, which a patient is lacking, or it can produce a toxin so that the disease cell is killed. So this is what we target for the cancer. Okay, the activate cells of the immune system so as to help in killing of the disease. So this is also for the cancer and the other neurological disorders. Now, what are the strategies? So what are the different gene therapy strategies? So first one is gene augmentation therapy. So what does it mean? In this, a simple addition of functional allele is used to treat inherited disorder. So see, this is something which is new. You need to understand this. See, if suppose a person is having inherited disorder, usually you don't have the treatment for that, right? Because it is from the birth and it is difficult. But nowadays, it is possible. Now, for this to be possible, the doctors should know the genetic compatibility. I have discussed about this, about the genetic compatibility. So whenever a couple gets married, he or she should also go for the genetic compatibility. Apart from the horoscope matching, they should also go for the genetic compatibility. So this will tell you whether in future, what is the probability that your kid, either it is son or daughter, is going to have the genetic disorder or not. Now, if suppose you still want to get married and you still want to have a kid, knowing that suppose out of 4, one might have this particular disorder. For example, albinism, let us say. So the albinism means the entire body will be very white in color. There will be no melanin, right? So let us say that they still agree and they want to get married and want to have kids, knowing that there will be certain genetic disorder. So what they can do is they can talk to their doctors, genetic doctors, they can talk, and with the doctors will then tell them what kind of injections or what kind of treatment is required. So at that level itself, at the zygotic level itself, they can go for this gene therapy, or once the baby is being delivered, that time also they can take the shots. Okay, so it will be like an injection only for the baby and the treatment can be started. So nowadays it is possible. Now, second one is targeted killing of specific cells. So it involves utilizing genes encoding toxic compounds. Those are called suicide genes or the prodrugs. So they are the reagents which confer sensitivity to subsequent treatment with a drug to kill the transformed cells. It is popular in cancer gene therapies, as I mentioned before. So anyway, whenever we're targeting any cells to die, we always remember mainly it is meant for the cancer. Now, how we do? So we have got the toxin gene. This is a vector you put into the cells. Now, every time you need to check for the disease cells and the normal cell also. Why? Because we need to do the analysis. We need to see whether it is only killing the disease cell or it is also killing the normal cell. So whenever you do any experiment, you need to have the control. So cells will be killed by the expression of the toxin, both. Now, if you take the prodrugs gene, again, the drug will be released and then the cells will be killed. Now, the antisense technology, this is very famous and many people are actually, most of the research which is based on the molecular biology, it is based on this antisense technology only. Okay, so it is the targeted inhibition of gene expression antisense technology. So this is happening at the RNA level. So when you talk about antisense, you are talking about the RNA level therapy. Okay, it is to block the expression of any disease gene which is harmful for a cell, particularly suitable for treating the infectious disease and also the cancer. See, again, it is cancer. So now you can understand like cancer has reached every corner of the world, and it has been not yet treated, even though it can be what to say controlled, but it cannot be treated completely. So many a times you must have heard that for example, osteosarcoma is there, so that is your bone cancer. So what people do, what suggestions can you give if I say that in the one of the leg it has got the bone cancer? So what is the best suggestion you can give in order to get rid of the cancer? Anyone? Pratima, what do you think? Ranjan, any idea?
No.
See, if I'm saying that this person is having bone cancer in this particular leg, okay, so there are treatment, chemotherapy, radiotherapy is going on, okay, but chemotherapy and radiotherapy does not give you 100% success rate. It will not tell you that 100% sure, yes, the therapy has cured the cancer. So what is the alternate method? Pallavi, what do you think? Don't be hesitant.
No,
No idea?
don't
Seriously?
know.
See, it's very simple. If I say that this person is having in this bone, the cancer is there, the best way to get rid of the cancer is to actually cut that much of the portion of your body.
Cut off. Yes. Yes.
I
Yes.
know it's a difficult decision for anyone to take because you will be handicapped for the rest of your life. So it's the option given to the patient, either go either go through the life have your life full of pain because chemo and radiotherapy weakens your body and your immune system, because of which every time you're in the pain, okay, and vomiting, nausea, dizziness, everything you will get. The other option is to get rid of that particular portion. The only problem is either you will be having this pain or the other pain, being handicapped. So yeah, so cancer actually destroys the life. So it's always, it's always good to have a good food habits and all. Very good lifestyle is very important. Okay, so coming back to the topic over here. So you have got the DNA. After transcription, you get the mRNA. Now mRNA, so we know that there are three possibilities in the case of antisense technology. So there is an RNase H, which can go bind to the mRNA and make a cut. So what will happen? No mRNA means no protein. Okay. The other is to go and make a cut into the ribosome itself. So again, no protein synthesis, right? Then you have got the risk. That is risk. This is for your sorry, RNA I. That is I stands for interference. So this is another technology which I will be taking later. So in this case also there is a complex which is called RISC, and it's just same. It is actually an RNA which goes and bind to your mRNA. That is double stranded RNA structure is formed, and then the endonucleases comes and makes a cut. So again there is no protein synthesis. So what is happening at the mRNA level? The RNAs are being chopped off, cleaved. Okay. So that's how you are also targeting. So these are the three strategy which comes under the antisense technology. Now, for gene therapy, as I was mentioning, we always make use of vector. Now, vector from where, which is easy to get, is the viruses, because viruses are easy to grow, okay, and they are very fast growing also. So from one cell, you can get millions of viruses, right? And yes, and the thing is that whenever we use viruses, we are making them attenuated. What does it mean? We are making them that able to infect the host but not able to cause the disease. Okay, so we are making use of the viruses as the vector. So adenoviruses are the DNA viruses. They're linear double-stranded genome of around 36 kilo base pairs. Now, when these viruses infect the host cell, they introduce their DNA molecule into the host. Now, what you have to remember, exam point of view, is that it is a DNA virus, linear, double-stranded, this much. It does not get incorporated into the genome of the host. See, you have got the adenovirus. It goes inside the host, okay, but it does not go inside or the DNA. DNA does not get integrated into the genome. Okay, now the linear double-stranded DNA genome remains non-integrated as an episome within the cell nucleus, and instructions in this extra DNA molecule are transcribed just like any other gene. So what does it mean? So whenever it enters the host, it will remain like this, and it will be having its own instructions to undergo replication, transcription, and translation for the gene to be expressed. Okay, is it clear? So as I mentioned before, there are two ways: either it remains over here only, or it gets integrated into the genome. So this is the case where the DNA of the adenovirus is not getting integrated into the host cell's genetic material. Is this slide clear? This is important.
Yes, ma'am.
Okay. Now, adeno-associated viruses. So these extra genes also do not replicate when the cell is about to undergo cell division. So what happened? The descendants of that cell will not have the extra gene. So what does it mean? Huh? The sentence meaning is what? See, in a cell, it is not going inside the genome. It will remaining like this, right? Now, so when a cell is dividing, what is happening? Two cells are formed, two daughter cells are formed. So it's not necessary that each daughter cell will have the adeno-associated virus. It's not necessary. There might be chance that only one cell is having and the other cell is not having. Okay. So because of this, whenever adenovirus based treatment is made, the regular doses have to be given. Are you getting the point? Huh? So the treatment with the adenovirus will require regular dose in order to add the missing gene every time, because whenever it is going to divide, one of the cell is not going to have and one will have. So in order to maintain this, the extra dose will be given. Adeno-associated viruses, they are from the parvoviruses. This is parvoviruses family and are non-pathogenic single-stranded DNA viruses. So they rely on the co-infection by an adeno or herpes helper virus to replicate. Okay. So these are the advantages and disadvantages of using the adeno-associated viruses as the source for gene delivery system. So you have got the retrovirus. We have discussed before once, adenovirus and adeno-associated viruses. So this is the overall summary of the advantage and disadvantages. So advantages. So retrovirus, it gets integrates into the host cell genome and it will give the gene expression. So whenever it is replicated, each offspring cell is also going to have, okay, but only few immunity problems. It's good. Now in the case of adeno, we know that it is not getting integrated. Now adeno-associated virus, it gets integrates into the host cell, okay, and it has got no immunity problem and it infects both the dividing and the non-dividing cells. So it's okay since it is not causing any infection to the host. So it's fine that if it is able to infect the both dividing and the non-dividing cells. Then coming to the disadvantages, it infects only the dividing cells. So in this case, what happens? Already, which are the non-dividing cells, it is not going to kill them, and it is random integration. So we cannot control its integration. Now, if the gene happens to be one regulating the cell division, uncontrolled cell division can occur. So it means what? Even if you're using retrovirus in order to treat one particular disease, there are chances that you are also inducing the cell of the patient to become cancerous. So there are many chances, many a times happens. So you have gone for some of the disease control. Okay, so you have gone for the treatment, but during the treatment, since you're using the retrovirus, there are chances that the patient who was not initially having the cancer might get cancer now. Okay, then disadvantage of adenovirus does not provide long term, and that's why you need to give the doses again and again. Then, in the case of adeno-associated virus, it is having the low carrying capacity. So, what do you mean by low carrying capacity? Low carrying capacity means, see, if a vector is this much is there, how much of the additional gene, that is gene of interest, what we use, it can carry. Okay, so maximum there are many vectors which can carry 25 kilo base pairs. In the case of adeno, it is around 4.7 only. So, it means what? A very small gene only can be targeted. So it has got low carrying capacity. So if suppose a vector is, if I'm saying that it is 25 kilo base pair, it means it has got high carrying capacity. Did you guys got the meaning? What does it mean, carrying capacity of a vector? Or should I repeat? See, vector is a, okay. So in the virus vector, there are many genes which is not required. That is for it to cause the infection and all. So that genes can be replaced by the other gene that is of our interest. So sometimes you can only add small gene, or sometimes you can add very big genes also. Now carrying this vector and introducing into the host cell, if it is able to replicate, it means what? This has got the low carrying capacity and this has got the high carrying capacity. That is to carry the extra gene which has been introduced by our action, that is by the doctors and the scientists. Okay. Now coming about the non-viral vectors. So this involves chemical and physical methods. It means what? We are not using any viral vectors here. Directly normal, it will be directly introducing the gene into the target cell. So in this case, it is the direct injection of the naked plasmid DNA, that is a particle bombardment. It is also called the biolistic. That is the gene gun. I hope you must have heard of gene gun. Or it will undergo then the receptor mediated endocytosis, and or we can have also the liposomes. And of course the nanoparticles, which is the latest now. So what do you mean by gene gun? So remember like how we were doing the micro injection when we were transferring the embryo, sorry, not transferring the embryo, when we were doing the embryo stem cell, right? So that time we were
Yes, ma'am.
using the micro injection. In the same way, this gene gun is also there. So the particular DNA sequence, it will be directly injected into the cell. Okay. So basically, this has been used for the muscular dystrophy problem. Now, what happens in the muscular dystrophy? The patient starts losing the muscles. So no muscle means what? Not able to walk, not able to sit, not able to move his hand and all. There is no muscle itself. So it's very thin, and they're not able to walk also. So it's very difficult for them. So they're losing the muscles. Now, so in this case, the gene gun was used, in which the DNA is coated onto a metal microparticles and delivered from a ballistic gun into the cells. So they are like the bullets. On the bullets, on the top of the bullets, you have got the DNA coated. It is like this, your gene of interest. And then like the bullets, using the gene gun, it has been transferred to the target cells. Right? So this is actually very simple. So it can cross the physical barriers like skin, muscular layer, for which it is used for the vaccination. And it is also called biolistic. So don't get confused that gene gun is different and biolistic is different. So both are same. So factors affecting the bombardments: nature of the microparticles, nature of the tissue, amount of DNA. So see, if you're using a very deep tissue, then the delivery of the DNA using the gene gun will be difficult. Okay. And then amount of DNA also, if you, of course, if you use less, there are chances that it might not reach the target cell also. So you always need to have the proper amount of the DNA to be used. Advantage, it is simple and safe. Disadvantage, poor efficiency. So this is what I was saying, that it is not necessary that every time whenever you use gene gun, it is reaching the target cell. And every time it's not necessary that whatever the gene you have targeted, it is also going and getting integrated into the genome. So it means what? Maybe all of your efforts are also going into uh, waste, because you have tried doing the biolistic approach, but the gene is not getting inserted into the genome, and hence no expression is coming. It is all getting wasted. Now liposome mediator. So guys, do you remember liposome? What is liposome from our previous lectures? So it is actually a double layer membrane, but into the spherical form. So whenever it is there, it is taken inside the water. What happens? Because of the hydrophobic stacking or interactions, the liposomes are formed. So this is, if you remember your previous classes, so you had your membrane like this, double membrane is there. Now if suppose you provide an aqueous solution you are providing, what will happen? This membrane will be folded like this, so in the center you are going to have the hollow. In the hollow space you are adding your DNA. Okay, so same thing. So when this lipid layer is taken, lipid bilayer is taken, DNA is taken, you mix it, liposomes are formed, and in the cavity your DNA is present. Now after that, this is also what? Your lipid membrane only, and in the target cell also you have got the plasmid membrane, so both are same, so it is easy for them to get merged. So the liposome and your targeted cell will get merged, and once they are getting merged, the DNA is delivered to the host cell. Is it clear, this method?
Yes, ma'am.
This is very simple actually. So they can be cationic as well as the anionic, that is depending on the charges they are using. So anion liposomes have got the negative surface charge and bind to the DNA on the inside. Of course, why inside? Because DNA itself has got the negative charge because of the phospho backbone, right? So they need to alter the structure of the DNA in such a way that it does not get repelled by the formation of the liposome. Okay, so advantage, no viral components are needed. So of course it is not pathogenic, no immunity problems will be there. Disadvantage is again the transfection is less and the expression, it is going to be very short. So whatever technique, whatever till now the techniques has been mentioned and what we have studied till now, it is very much clear that each technique gives the idea that it is possible to do. But every time, what is the disadvantage you're seeing? That it has got the poor efficiency. And because of this poor efficiency only, till now we are unable to cure many of the diseases. Like for example, cancer. Okay. So yeah, that was all about the gene therapy. Thank you guys. And with this, I would like to conclude that your animal tissue culture portion has been completed from the syllabus. Thank you for watching.